The Concise Encyclopedia of Genomic Diseases is a book whose intended audience includes physicians, biological scientists, and students of medicine, biology, molecular biology, genomics, and bioinformatics. The major objective of this work is to educate the aforementioned medical professionals for the sake of genomic disease prevention, as well as for understanding the genomic basics of clinical knowledge. This book will enable the reader to understand the reasons why genomic diseases emerge, the demographic groups in which they prevail, and the strategies needed to prevent them. There are a few concepts that will be used throughout the book. At the center of all of them are genomic diseases. Other concepts used in this book include product of consumption alteration, dissociative mating, diversity, meiotic non-disjunction event, anticipation events, trinucleotide repeat expansion, de novo disorders, and time of DNA alteration origins, among others. This book has an introduction, 5 chapters, and a conclusion. It has 2 volumes, the first of which contains the introduction, Chapter 1, Chapter 2, and most of Chapter 3. Chapter 3 is mainly the clinical genomic description of each condition and constitutes around 60% of the entire project. Volume 2 includes the remainder of Chapter 3, Chapter 4, Chapter 5, and the conclusion. The introduction addresses the genomic disease from the following perspectives: historical, statistical, ethnic, religious, and geographical, ethnicity, race, and population, nationality, ecology, and other miscellaneous factors. About 200,000 years ago, Homo sapiens emerged in Africa. Modern humans were on the verge of disappearance 71,000 to 74,000 years ago. During the volcanic winter event, a genetic bottleneck was created, at which time only 2,000 to 15,000 people survived. Some genomic alteration events took place before the development of agriculture more than 10,000 years ago. Nowadays, these traits produce diseases such as cystic fibrosis, factor V Leiden, and Friedreich ataxia. At the beginning of the agricultural times, many genomic traits were already present. 10,000 years ago, the worldwide population was 4 million, which is roughly the number of people living in Minnesota today. 2,000 years ago, the worldwide population was 170 million, which is about the population of modern-day Brazil. Today, the human population has increased to 17 times the level that it was less than 500 years ago. Multiple alterations have occurred during this period. For example, the major allele that causes cystic fibrosis is the result of a DNA alteration, the CFTR, which occurred 10,000 to 52,000 years ago, and mainly affected Europeans, Middle Easterns, and Western Asians. Another example, deafness 1A, is caused by mutations in the GJB2, which occurred 14,000 to 17,000 years ago, mainly affecting Europeans. A more recent example of alteration is mucolipidosis 2, or one-cell disease, which has been reported in more restricted ethnic groups. Among these groups, French Canadians are affected by the actual DNA alteration that happened in the Mediterranean region over 2,000 years ago. The first chapter describes genomics in the past, present, and future. The goal of the chapter is to adapt Mendel's law of inheritance into the contemporary genomic field to prevent genomic diseases in the future. Chapter 1 discusses, number 1, modernized Mendel's inheritance laws or post-genomic revolution laws of product of conception. Number 2, classification of product of conception alteration and clinical examples. Number 3, parent of origin of de novo alteration. Number 4, prevention of genomic diseases. The next 2 slides describe a concept that is highly encouraged by this book: conscious reprogenomic programs. These have been targets of criticism due to certain atrocities in the 20th century, such as compulsory sterilization, enforced abortions, enforced pregnancies, marriage restrictions, and the like. Unlike the 20th century approach, according to which marriage restrictions were based on race, this new approach of conscious reprogenomics encourages dissociative mating based only on known disease traits. It also eradicates the occurrence of homozygosis in lethal alleles. 1 in every 10 marriages worldwide is between first or second cousins. These are predominantly common in the Middle East, where they account for over half of all marriages in some nations. On the other hand, it is harder to decrease the prevalence of polygenetic or complex diseases, since the identification of genomic risk factors is still limited and does not follow a clear-cut pattern of inheritance. Avoiding advanced parental age is one of the three most important ways to prevent genomic diseases. In most modern societies, no measures have been taken to avoid late parenthood. For example, in the United States, the average age of first pregnancy has increased by 4 years since the early 80s. The data concerning this issue is limited. The data regarding parental age should be broader in the sense that it should not only focus on the first pregnancy, but more on the age of the last pregnancy. Data regarding the male parent is equally or perhaps more important than data on the mother, since males often reproduce at an older age. Fortunately, data records of the age of male parents are limited. The study suggests that data on males' age at the time of reproduction should be taken into account in de novo genomic disease studies. The second chapter analyzes the strategies for understanding pillars of genomic origin in diseases, using not only correlation between alteration of DNA information and disease, but also criteria of inclusion and exclusion. It's sometimes hard to determine the level of genomic component in a disease, but several studies, including family and twin, have tried to address this question. Sometimes environmental and behavioral factors provide a better explanation for a disease than genomics. Chapter 2, Strategies to Understand the Pillars of Genomic Origin in Diseases. The mechanisms detailed in the book include correlation between alteration of DNA information and disease, set of people where variation in DNA correlates with the disease, dissecting the mechanisms of disease, classifying disease into groups, identifying alteration in productive conception, establishing origin of the disease, making use of exclusion and inclusion criteria based on twin studies, first-degree relatives, and familial risk. There are many wrong assumptions regarding inheritance in genomics. One of the most common assumptions is that the traits are mainly dominant. This is based mainly in common sense information. If your father had a heart attack or your mother had breast cancer, you have the same probability of suffering those conditions. But these assumptions are generally wrong. The third chapter examines the classical method of understanding clinical syndrome in genomics. Most of the diseases, from susceptibility to infection to cancer or cardiovascular disorders, are rooted in our genes. One of the objectives was to look at all the genomic information on clinical syndromes in order to see the pattern to comprehensively understand the clinical genomic aspects of diseases. This chapter introduces a new way of thinking about genomic diseases with respect to their corresponding genomic data and their correlation with other diseases. The chapter has two main parts. The first part discusses in detail the disease groups and classification methods for a select group of diseases. The second part of the chapter concisely describes all known genomic diseases and their traits. For instance, by studying neurology, one can focus only on the neurologic disease. However, by overlooking the genomic information that is in the same locus, the traditional method will miss important clinical information. Instead, the new method will avoid dropping important clinical and genomic information. In considering the genomic diseases and conditions, it is important to reference back to the OMIM database, which is the cornerstone for this chapter. However, some information in this database is slightly outdated, since many genes have been renamed and the database hasn't been updated. The rules and coding mechanisms that I have used to help the reader better engage with and understand the disease classifications are presented in the section Classification Codes. This chapter has been further divided into two sections. The first section deals with the approach to creating a comprehensive database, and the second section deals with the clinical descriptions of genomic diseases. This part of the book is divided into 18 subchapters, and the goal is that every human disease has to be accounted for in at least one of the subchapters. The three largest subchapters are congenital malformations, deformations, and chromosomal abnormalities, diseases of the nervous system, endocrine, nutritional, and metabolic diseases. Not every cancer is included, since the primary focus is product of conception alteration, not the somatic changes. The third subchapter describes 4,000 diseases, but there are certain diseases that are not included. Among these diseases, I selected 11 for further examination. Cystic fibrosis or mucoviscosis, a disorder characterized by the production of mucus secretions with abnormal viscosity and sweat with high salt content due to abnormal transport of chloride. This disease usually starts at birth with meconium ileus or during early childhood, but there are also late onset forms. Cystic fibrosis is caused by autosomal recessive loss of function mutations in the CFTR gene, which encodes the cystic fibrosis transmembrane conductance regulator. This condition is an example of the loss of function mutation during early migration trait. These pathogenic alleles started to appear 10,000 to 52,000 years ago, most likely in Europe, the Middle East, and Western Asia. Cystic fibrosis traits are mainly due to founder effect and endogamy. The product of conception alterations are defined as occasional de novo autosomal recessive syndrome, or AR plus KDN, with unknown prevalence of de novo polymorphism. This is the most common life-limiting autosomal recessive disease among people of European descent, except the Finnish. Genomics follows statistical rules, and the allele frequency can predict the disease frequency. For example, if a Caucasian American man with allele frequency of 1 in every 50 reproduces with a Caucasian American woman with allele frequency of 1 in every 50, this results in 1 in every 50 squared, or 1 in every 2,500 Caucasian Americans who are at risk of having cystic fibrosis. This is one of the reasons why conscious reprogenomic programs are required. While certain diseases are often present in the same ethnic group, the color of the skin or any general characteristic of an ethnic group is not the best predictor of the prevalence of cystic fibrosis. For example, there's a group of people in Finland that has 20-fold less frequency of the disease than a neighboring group. This emphasizes that although the Finns and Irish are white Caucasians, they do not have a similar prevalence of cystic fibrosis. Alpha thalassemia, a disorder characterized by a reduction in the amount of hemoglobin in the red blood cells. This form of thalassemia involves two subtelomeric genes, HbA1, which encodes alpha-1 hemoglobin, and HbA2, which encodes alpha-2 hemoglobin. Alpha thalassemia is a digenomic disorder characterized by impaired production of 1, 2, 3, or 4 alpha globin chains. There are two main different genomic types, or subtypes. First subtype is common in sub-Saharan Africa, the Mediterranean basin, and the Middle East. It is characterized by a single monoallelic deletion of an alpha globin gene. The alpha thalassemia minor affects 30% of African Americans. Second subtype is rare, affecting individuals from Southeast Asia, producing serious clinical disorders of hemoglobin H disease and hydrops fetalis syndrome. This subtype is characterized by double biallelic deletion of an alpha globin gene. Among Southeast Asians, the frequency of the biallelic deletion trait is estimated to be 1 in 25 individuals, which means the frequency of the condition is 1 in 2,500. Sickle cell anemia, or drepanocytosis, a form of anemia characterized by hemoglobin polymerization leading to a rigid erythrocyte with a sickle shape and resulting in vaso-occlusion. Symptoms usually start during childhood. Sickle cell anemia occurs more commonly in individuals from equatorial Africa. It also affects individuals from the Mediterranean, India, and Middle East. The geographical area is shared with malaria, providing a fitness advantage. The sickle cell trait is originated around 2,000 years ago. It is caused by biallelic polymorphism in the HBB gene that causes HBS. There are other less common forms of sickle cell anemia, which include sickle hemoglobin C disease, sickle beta-0 thalassemia, and sickle beta-plus thalassemia. There are different geographical origins of the sickle cell gene alteration. These variants are known as Benin, Bantu, Cameroon, Saudi Asian, and Senegal. The genomic disorder is due to the polymorphism of a single nucleotide from a GAG to GTG codon mutation, resulting in glutamic acid being substituted by valine at position 6 of the beta-globin protein. In the United States, it is a rare syndrome affecting 1 in 5,000. Among African Americans, the carrier frequency is 1 in 12.5 individuals, which means the frequency of this condition is 1 in 625. Huntington's disease, or Huntington's chorea, a disorder characterized by chorea, cognitive decline, incoordination, dystonia, and behavioral difficulties. Huntington disease usually appears in the third or fourth decade of life. It is caused by autosomal dominant CAG expansion of the HTT gene, which encodes the huntingtin. The expansion of CAG creates a polyglutamine tract, which produces an adverse gain-of-function mutation due to increased aggregation. The HTT gene is located in the subtelomeric short arm of chromosome 4 at 4p16.3. Huntington disease can be inherited from the mother or the father, but the main phenomenon of anticipation in this case is due to CAG expansion that occurs during spermatogenesis in the paternal allele. The product of conception alteration in this condition is AD plus ANT. Trinucleotide expansion occurs in paternal allele and contraction in maternal. At least one allele that matches with the trinucleotide expansion in Huntington disease has an origin in Europe from 4,700 to 10,000 years ago. The pathogenic allele in Huntington disease is an example of DNA alteration during early migration trait, founder effect, parental advancing age, subtelomeric condition, gain-of-function mutation. The disorder seems to be more common in several regions of the world, including Scotland, Wales, Sweden, Tasmania, and in the Lake Maracaibo region of Venezuela. Achondroplasia, a disorder characterized by short stature caused by rhizomelic shortening of the limbs, characteristic facies with frontal bossing and midface hypoplasia, exaggerated lumbar lordosis, limitation of elbow extension, genuvarum, and trident hand. It is the most common cause of dwarfism and is caused by monoallelic mutations in the FGFR3 gene, which encodes the fibroblast growth factor receptor 3. In 98% of cases, a G to A point mutation at nucleotide 1138 of the FGFR3 cDNA causes a glycine to arginine substitution. This monoallelic condition occurs as a de novo mutation in 75% of cases and is inherited in an autosomal dominant pattern in the rest. Gain-of-function mutations in this gene cause a constitutively active form of the FGFR3 gene, which leads to severely shortened bones. The FGFR3 gene is located on the subtelomeric short arm of chromosome 4 at 4p16.3. Advanced parental age has been associated with significantly increased rates of de novo achondroplasia among offspring compared with younger fathers. Achondroplasia is estimated to affect 1 in every 25,000 individuals. Duchenne muscular dystrophy, a disorder characterized by muscle degeneration, difficulty in walking, breathing, and death. Symptoms frequently start to manifest before the age of 5, and most patients become wheelchair dependent by 12 years of age. Duchenne muscular dystrophy can be caused by X-linked recessive mutations in the DMD gene, which encodes the dystrophin. This rod-like cytoskeletal protein is found at the inner surface of muscle fibers. Mutations within the gene are inherited in 76% of cases and occur de novo in 24%. The product of conception alterations are defined as X-linked recessive syndrome plus 30%. Duchenne muscular dystrophy is estimated to affect 1 in every 3,600 newborn males worldwide. Hemophilia A disorder characterized by lower blood plasma clotting factor levels than are required for a normal clotting process. Patients with severe hemophilia have 20 to 30 yearly episodes of spontaneous or excessive bleeding after minor trauma, predominantly in joints and muscles. Hemophilia A is caused by X-linked recessive mutations in the F8 gene, which encodes the coagulation factor 8. These mutations are inherited in 70% of cases and occur de novo in the remaining 30. The gene is located on chromosome X subtelomeric long arm. Hemophilia A belongs to different groups including subtelomeric disorder, de novo disorder, and X-linked recessive disorder. Hemophilia A represents 80% of all hemophilia cases. This condition is the most prevalent form of the disorder, affecting 1 in 6,000 male births worldwide. Glucose-6-phosphate dehydrogenase deficiency, or FAVism, a trait characterized by a hemolytic reaction to the consumption of fava beans. Patients might suffer from non-immune hemolytic anemia in response to the consumption of fava beans, infection, or exposure to certain medications or chemicals. Glucose-6-phosphate dehydrogenase deficiency is the most common enzyme defect affecting more than 400 million people worldwide. This disorder affects mostly males since it is an X-linked recessive syndrome. This trait confers protection against malaria caused by Plasmodium falciparum. This trait and malaria also share the same endemic areas. Glucose-6-phosphate dehydrogenase deficiency is caused by X-linked recessive mutations in the G6PD gene, which encodes the glucose-6-phosphate dehydrogenase. This gene is located on the subtelomeric long arm of the X chromosome on band XQ28. X-male syndrome or De La Chapelle syndrome or 46, XX sex reversal 1. a disorder characterized by true hermaphroditism with ambiguous genitalia. This condition results from an unequal crossing over between the subtelomeric region of chromosomes X and Y during parental meiosis, resulting in an X chromosome that contains the normally male SRY gene. When the spermatozoid fertilizes a normal ovocyte, the product of conception alteration results in an XX male. These products of conception alterations are defined as chromosome XY recombination. XX male syndrome is estimated to affect 1 in 5 in every 100,000 individuals. Down syndrome, or chromosome 21 trisomy, a disorder characterized by the presence of an extra copy of genomic material on either the whole or a part of the 21st chromosome. Individuals with this condition have an average IQ of 50, physical growth defect, and particular set of facial characteristics. Patients with chromosome 21 trisomy also suffer from congenital heart disease, neoplasia, thyroid disorders, gastrointestinal disorders, infertility, neurological disorders, ophthalmology disorders, and hearing loss. This is the second most common autosomal trisomy after chromosome 16 trisomy. The incidence of Down syndrome has been rising in the last decades, and this condition is statistically more common in children born to older parents. It is estimated to affect 1 in every 690 births in the United States. Advanced maternal age is widely recognized as increasing the risk for chromosome 21 trisomy. For instance, the risk of having a live born with chromosome 21 trisomy at maternal age 30 is 1 in every 1,000, whereas at maternal age 45 it is 33 in every 1,000. Down syndrome occurs in all sets of people and all ethnic groups around the world. Fragile X syndrome, Martin-Bell syndrome, or Escalante syndrome. A disorder that is the most common known single gene cause of autism and most commonly inherited cause of intellectual disability. Fragile X patients exhibit intellectual disability and physical characteristics such as elongated face, larger protruding ears, stereotypical movements, social anxiety, and macroorchism. Fragile X syndrome is caused by X-linked monoallelic mutations in the FMR1 gene, which encodes the Fragile X mental retardation protein 1. Individuals with Fragile X syndrome have over 200 CGG repeats in the FMR1 allele. These products of conception alterations are defined as classic X-linked dominant syndrome plus anticipation. The origin of this elongated allele is mainly maternal, since it takes many generations to have a full Fragile X syndrome. Male carriers pass on their pre-mutation to all of their daughters without increasing the CGG repeat length of the FMR1 gene during meiosis. However, females with a full mutation are able to pass this full mutation on to the next generation. In addition, the length of the CGG repeat frequently increases during meiosis in female carriers. Fragile X syndrome is estimated to affect 1 in every 3,600 males and 1 in every 4,000 to 6,000 females. There is ample evidence to support the formation of different groups of diseases such as SNP syndrome, subtelomeric syndromes, trinucleotide syndromes, insertion syndrome, deletion syndrome, and de novo syndrome. This slide discusses the previously described conditions in which 6 out of 11 disorders are subtelomeric. Most conditions are caused by SNP syndrome. This slide discusses the previously described conditions in which 2 out of 11 disorders are due to trinucleotide repeat expansion. The classical example is Huntington disease due to CAG trinucleotide repeat expansion. Another example is Fragile X syndrome due to CGG trinucleotide repeat expansion. The fourth chapter illustrates the reverse method of understanding genomics in clinical syndrome. It provides ample evidence to support the formation of different groups of diseases, several groups of diseases such as SNP syndrome, subtelomeric syndromes, trinucleotide syndromes, insertion syndrome, deletion syndrome, and de novo syndrome are based on DNA alteration. Since not all the SNPs or mutations produce a disease, this chapter focuses on DNA alteration rather than SNP or mutation. Because there are 2,300 gene-producing diseases and 54 million SNPs, the only manageable study to conduct at this point is the data from the gene-producing disease when available. Chapter 4, Genomics and Clinical Syndrome, discusses time of DNA alteration origin, parental age and the origin of DNA alteration, genomic disorders related to structure in genes, and/or chromosome, disorders related to structure in chromosome, disorders related to genomic silencing and or imprinting, disorders related to oligomeric or multimeric protein dysfunction. The origin of pathogenic alleles and cystic fibrosis has been traced back to DNA alterations that occurred 10,000 to 52,000 years ago. The origin of pathogenic alleles in thalassemia has been traced back to DNA alterations that occurred over 50,000 years ago. There are 5 different haplotypes for sickle cell anemia. The origin of the Bantu pathogenic allele in sickle cell anemia has been traced back to DNA alterations that occurred over 2,000 years ago. Huntington disease, 4,700 to 10,000 years ago. Fragile X syndrome or Escalante syndrome, few generations ago. Duchenne muscular dystrophy, few generations ago. In general, the DNA alterations for hemophilia A occurred a few generations ago, but this condition has been known since the 2nd century AD, when it was described in the Talmud. Achondroplasia, de novo alterations. Down syndrome or chromosome 21 trisomy and XX male syndrome or De La Chapelle syndrome are examples of de novo disorders. The origin of allele or disease can be classified into 3 groups: ancestral, early migration, and de novo. According to the size, affected DNA alterations can be classified into 3 groups: short genomic variations, large genomic variations, and chromosomal non-disjunction alterations. These alterations in few nucleotides could further be categorized into 4 groups: single nucleotide polymorphisms, multinucleotide polymorphisms, microsatellite markers, or short tandem repeats, and short deletion and insertion polymorphisms. Large genomic variations. This is a group of structural variations which are larger than SNPs and smaller than chromosomal non-disjunction alterations. Usually, a large genomic variation affects a sequence length about 1 kb to 6 MB. The variation consists of deletions, duplications, copy number variants, translocations, inversions, insertions, ring chromosome formations, isochromosome formations, and complex genomic rearrangements. The designation of a large genomic variation does not imply an association with genomic disorders. Nevertheless, there are variations that are associated with genomic disorders. Large genomic variations are most difficult to detect than SNPs. In people with no known genomic disorders, around 5% of the genome affecting 800 genes is designated as structurally variant. The 5th chapter describes the biological method of understanding genomics and clinical syndrome affecting the organelles. This includes the nucleus, the biological membranes, the cytoskeleton, extracellular matrix, other organelles and their functions, miscellaneous categories, and the conclusion. Conclusion. The Human Genome Project is undoubtedly one of the most thoughtful research efforts in human history and now presents great opportunities for practical application. The author of this book is concerned with understanding the social implications that emanate from the Human Genome Project. On the other hand, the implications are still mainly theoretical rather than practical. The promise of this work lies in opening the door towards more practical results in the genomic field. We have yet to implement a program to effectively teach that lower parental age at the time of conception will prevent genomic diseases in future generations. On the contrary, in the same period of time in the United States, there has been a rise in the average parental age during conception. This illustrates our failure to effectively apply the lessons we have learned from genomic sequencing. Based on studies provided in this book, many de novo disorders are related to advanced parental age, and many autosomal recessive disorders are related to reproduction amongst individuals with similar genomic backgrounds. Genomic bottleneck and founder effect can be diminished if reprogenomic programs are implemented to decrease autosomal recessive disorders. In practical terms, this book emphasizes that it is important to assess the future product of conception. Widespread preconception counseling and education are required and must be fulfilled, so our society decreases the advancement of de novo disorders and traits. The author of this book does not encourage the traditional geopolitical separations according to nationality and race. In fact, there is almost no reference according to nationalities in the entire book. Genomics is a cosmopolitan conception, since all human ethnic groups belong to a single group of people based on a pooled genomic makeup. We may have not realized what our society risks by continuing the trend towards delaying parenthood. The reprogenomic consequences of postponing parenthood, as illustrated in this volume, could be devastating to society. For both males and females, the biologically optimum period for reproduction is between 20 and 30 years of age. The Concise Encyclopedia of Genomic Diseases argues for a radical social change in our modern time.